Hello, class, and welcome to week 5. Today, I want to go over lab video with you to help you with this lab. For this lab, we're going to do a microscope lab, and this is just to get you familiar with working with the microscope because next week, for week 7 and week 8, no, sorry, for week 6 and week 7, you're going to be doing a two-part lab in which you're going to be looking at microscope slides of your water samples. And so this week is just to get you familiar with the microscope itself. And so you should have gotten a microscope with your lab kit. Okay, should look similar to this. Also, you should have gotten a yeast specimen. Okay, so a little vial of yeast. You should have also gotten a brine shrimp specimen. Okay, and then you should have gotten 3 empty specimen bottles. I dropped one, but you should get 3 of those. You should get a packet of slides. There should be 3 slides that are prepared. Okay, in my prepared slide, I got wool. I got frog blood smear, and I got an onion bulb. And I'm not sure if all the prepared slides are the same. So if you didn't get the same prepared slides, that's okay. And then you should have gotten, um, there should be some blank slides in there. Okay, you'll use some for this week, and then you use some for next week's lab. And then you should have gotten 8 labels and slide covers, a little pack of that. All right, you should have gotten a graduated cylinder and then a hatchery. Okay, that's for the shrimp. You should have gotten a specimen slicer. And this is your plastic transfer pipette, some forceps, and a magnifying glass. Okay, and so all of those should have come with your kit for the lab. All right, so what I want to do is I want to go over in your lab man I'm sorry, in the little the microscope manual, okay? I got mine from online, but I want to go over the first part of the manual just goes over how to operate the microscopes. Make sure you read through those. And then it talks about ideas for observations. So I'm going to move that over here. And I want to share my screen. Okay, share. All right, so we're going to go over some of these because some of these are in your lab that you're going to be doing. Okay, so in your lab, and I'm going to show you where to find the information for your lab. Okay, so I'm going to move this over here because I want to go over it with you through this. Okay, so for this week's lab, so first of all, let me show you this and then I'll show you the other thing. All right, so for this week's lab, this is the fundamentals of microscopy data sheet. So each week, I always like to go over what you're going to be doing for the lab and how to do your lab report. So this is actually how you're going to set up your lab report. And so I want to go over that first with you because in here are the observations you're going to be doing with your microscope. So the first thing is your introduction. You always remember you start off with your introduction. You have of course, you're going to do your title page. Okay, that's always first. And then you start your introduction. And so you want to give information about the microscope and the uses. Also include a picture of the microscope with labels for each part. See lab instructions. And so there is in the lab instructions, which is a PDF. And I'm going to show you where to find that. You have an actual picture of the microscope with the label. Okay, and so let me show you that. All right, so this picture, figure 1, this is in your PDF, and I'm going to show you where to find that in your Canvas page. But this picture you want to include in your lab report. Okay, so it doesn't have to be this large. Okay, you see how you can click on the picture, you can edit the image. Okay, and so if you edit the image, it's gonna open the image in editor. Okay. And so once you have edited, because you want it to be smaller, and so you can edit how you want it viewed if you want to resize it or crop it or whatever. All right. And then once you've done that, you can right click on it and it should be able to copy and paste. Let me see. Image delete, export. No. Let me see. Maybe when you edit it, maybe they give you a chance to copy and paste because you want this image to be in your lab reports. Let me see if you're able to copy and paste this. All right. So say we make that smaller. All right. So if we make that smaller, we apply changes. Let me see. Mm, image check. Convert. I think they're gonna make you do the trial. Okay, no. All right, so you might not be able to do that. So you might have to, let's see, I wonder if you can highlight. Trying to think of ways you can do this. All right, if you figure out a way, I can't find a way right now. But if you find a way to just copy and paste, oh, there we go. Copy image. That was easy. All right, and then you want to minimize that. All right, so then you would want to go to your Word document and you want to paste it. That's not what I want to do. I want to go home. Then I want to paste it. All right, there we go. All right, so you just simply copy and paste it. And then you in here is where you can make this small. Oh, nope, didn't want to reverse it. Okay, whatever I did to reverse it, I don't want to do that. There we go. All right. So you do want to make that smaller because you don't want that to take up your entire lab report, but you want that to be included in your introduction. All right. So let me take that out. All right. So then that is your introduction part. Remember, your introduction needs to be in your own words. Very important. The materials, all the materials that I just showed you that you're including for this lab. The other things that you're including, you're doing mold. And so I'm going to show you where to find the information about how to grow your mold. Okay. Your household item, you're going to put that, whatever you choose as your household item, that's going to go in materials. Your brine shrimp, your salt and your sugar, your pollen sample, your feathers or hair sample, your fiber sample. Okay, you can use, if you had the wool, okay, as your prepared slide, that could be your fiber sample. Okay, if you did not have wool, you can use something that's cotton, like you could use a piece of cotton material or something like that. That would be your fiber sample. And then your pond water sample, okay? And so you would get some water from a pond, or if you have a lake nearby, um, you want a sample of that water. And then a stain tissue section, okay? So you want that. All right. And so then all of that is going to go into your materials. your procedure, what you did, okay, for the lab. You want to put that in your own words. And remember, materials and procedures, those two sections need to be written in sentences and paragraph form, okay? I don't want a list, okay? Don't want just a itemized list. It needs to be in paragraph form for both of those sections. All right, so the observations is similar to our results section that we use for most of our labs. So the observations you're going to be using, you're going to be taking pictures. Okay, and in your lab, it does tell you how to take pictures using your microscope. There's a video on how to do that. I'm going to show you where to find that because that's going to be important so that you get great pictures of your different microscope images. Okay, so you don't have to, if you use that, you don't have to do drawings. Okay. And so if you take the pictures, then those will suffice. If you're not able to take clear pictures, then you would have to draw what you see, okay? Hopefully you're able to take good pictures, okay? So you don't have to draw, all right? Unless you like to draw. I personally am not an artist, and so my drawings would look horrible. So I would prefer the pictures, but if you prefer to draw, go for it. All right. And then discussions. So, like all discussions, there's going to be questions. These are questions you're going to answer in paragraph form, okay, for your discussion section. Then the last page is going to be your references. You are going to reference, okay, you have a couple of things. So you have this data sheet, you're going to have the instruction sheet that I showed you, that PDF instruction sheet. You're going to have that. All right. And then you're going to have the video that's going to show you how to take pictures of what you see in your microscope slides. So you have a few references that you're going to use, and any other references that you would like to add, make sure they are in APA style. Okay. So how do we get all of these different photos? So let's go to that next. All right, so this is the manual that came with your microscope. If you did not get this manual with your microscope, let me know, okay? Because I do have this PDF form that I got off the internet because I cannot find my manual. And so, but what you're gonna do is the crystals, these are the few grains of either dry salt or sugar on a slide. So you just place on the slide and you observe it. So then you want to mix the salt with a spoonful of warm water in the granulated cylinder, graduated cylinder. So I get my graduated cylinder so you can see that. It fell on the floor. Okay, so you're going to use the graduated cylinder and you're going to mix salt with a spoonful of, I'm sorry, salt with this, yeah, a spoonful of warm water in the graduate cylinder. Then use the plastic transfer pipette. That's this one. Okay. And you're going to place a couple of drops on a blank slide, let it dry, and observe the recrystallized salt or sugar. All right. And so that is how you're going to view it. Once it's recrystallized, then you're going to look at it on the slide. Okay. Then you want to shake some grains of pollen from a flower onto a blank slide and then observe that. All right. So for your crystals, you can see the crystals just as crystals, and then you can watch them once you've recrystallized, okay, and see the difference. Remember, you're taking pictures of all of these. All these pictures need to be included under your observations, okay? And then hair, include pet hair or pieces of feathers can be observed. Try comparing hairs from various animals. So if you have pets at home, you can get some of their hairs. Pets are always shedding their hair. Grab a hair, put it on a slide, okay? Place a small piece of tape on each end of the hair to hold it on the slide. Okay, that way you can see it better, all right? Then you want to observe fibers. Okay, you can use paper, you can use newspapers, color in comics, compare threads of fibers from different types of fabrics. So all of these you can observe those different fibers. Okay, remember we're taking pictures of all of this. With the pictures, when you're putting them in your lab report, very important that you label them. Okay, label what those pictures represent. All right. Stagnant water from a pond will contain live organisms. Place a drop into the well of a blank slide for observation. Okay. And when you're gathering like some pond water, you have these specimen vials. You can always put your pond water in these specimen vials to transfer them. And that goes for your pollen and all that other. Okay. All right. So many specimens may look transparent under the microscope. It's common to stain them to make the cells show up better. Iodine is a common stain. If you use iodine to stain it, okay, you can get iodine. Comes in a little bottle, tincture of iodine. You can get it from Amazon. You can get it from um, most drugstores, pharmacies. They have iodine, and you can use that as a stain. Okay. And you can use tweezers to pick up your stained specimen and allow the stained specimen to dry before mounting it onto the slide. All right. So that's a helpful tip. All right. So then your mold growth, all right, is the next thing you're going to do. So you're going to get some water, a paper plate, and a piece of bread. So you're going to place the bread on the plate, drop a little water on the bread to make it damp. You don't want to soak the bread. Then you want to place the bread in a cool, dark location so a cabinet is perfect, and observe the bread daily. Do not eat it. We do not want to eat our molded bread. Once the mold has formed on the bread, place the plate with the moldy bread under the microscope. Okay, if your plate is too large, okay, because we do have a small microscope, you can take a piece of the bread, okay, take your forceps and pinch off a piece of the moldy part and put it on a slide, put that under your microscope. That's perfect. All right. And then your brine shrimp. All right. So you have your hatchery, okay, which is shown here. You have your brine shrimp and you have your yeast specimen. All right. And so you're going to start with 2 tablespoons, okay, of water, okay, or 1 fluid ounce of water. All right. And then you want this water to be bottled or filtered. Okay, so just grab some bottled water, or if you have a filtering system that you use at home, get some filtered water and in a small container. Tap water can be used if you don't have filtered or bottled water, but it should be left to sit for approximately 24 hours. This is to disseminate any chemicals that are in the water that may hinder your shrimp from hatching. All right. So you're going to add a half teaspoon of sea salt to the container and stir until all of the salt is dissolved. The water should be room temperature, not hot or cold. Very important. Add a pinch of brine shrimp eggs to the container, okay, and place the container in a bright location, but not in direct sunlight. One option is to place the container directly under a desk light or somewhere similar. You can also put some salt solution along with a few eggs into each of the 3 compartments in the hatchery. All right. And then leave the lid slightly off or completely off for the brine shrimp eggs to receive oxygen. The ideal temperature is 70 to 75 degrees Fahrenheit, which is room temperature. At this temperature, the shrimp will hatch in about 2 to 3 days. So because it takes 2 to 3 days for them to hatch, you want to make sure that you don't wait till Sunday to try to do this lab. You want to give yourself at least 3 days for your eggs to hatch because you want to be able to observe it under the microscope. Okay. So if the water in the glass evaporates, repeat the solution of incubation without adding brine shrimp eggs and pour that new salt solution into your existing experiment. All right. So once the shrimp have hatched, You can observe them by using your hatchery and the microscope. So the hatchery is small enough that it will fit under the microscope so you can see the shrimp in the hatchery. Okay. You can review the shrimp in the hatchery using the magnifying lid. Okay. Placing the hatchery on the dark background may work best. All right. And to view the shrimp under the microscope, use the plastic transfer pipette in to take a tiny drop of water and shrimp from the hatchery and deposit it onto the center well of one of the blank slides. Place a round cover slip over the drop of water and put the slide into the microscope tray. With your naked eye, you should be able to see if you captured any shrimp. If not, repeat the steps until you do. Okay? And then it goes on to tell you the magnification and so forth. How to feed and clean the brine shrimp. So for the brine shrimp to survive, they must be fed once a week. Take a pinch of the yeast. Okay, so you have the container of brine shrimp. And you also have the little vial of yeast. Okay. So you take a pinch of the yeast from the specimen vial provided and deposit it into the brine shrimp container. Make sure not to overfeed your brine shrimp. And it talks about overfeeding and so forth. I'll let you read through that. All right. So those are the things that you want to look under your microscope. Remember, you're going to take pictures. And so I'm going to go over with you how to do that. All right. So first of all, this is your Canvas classroom, and this is where you find those instructions that I just went over. And so you have your data sheet and your PDF. The data sheet is where I showed you where you have all the information that you will need for writing up your lab report. This is your data sheet, okay? And then the other thing is the PDF. And so for your PDF, let me bring that up. Okay, so this is your PDF. In here, it's going to show you how to take pictures. Let me see. It goes through how to operate the microscope as well. But in here, let me find it. Okay, here we go. All right, so how to take pictures through the microscope. This is a video that I'm gonna bring up, and it's gonna go through how you angle the camera so that you can see and take pictures using just the camera on your phone, okay? So make sure you look through that so you know how to get those pictures so that you can add them to your lab report. Once you have completed your lab report, just as always, there is a Dropbox, okay, where you're going to submit your lab report for this week. If you have any questions, before I go to questions, let me go over the cleanup, okay? So disposal and cleanup. So you're going to return your microscope slides to the storage box, place them with the kit. You want to cover the microscope or return it to the box and store for future use. If you have grown or and observed bread, mold, or brine shrimp during your assigned activity, which you have, you may kill either with bleach, which is an excellent disinfectant. Be sure to wear your PPE, which is your gloves, your apron, and your safety goggles. And then whenever working with bleach, and ensure that you are in a space with adequate ventilation, okay? Because we want to make sure that um, it doesn't affect your breathing, okay? That bleach is a very strong and potent. So for your mold, you want to prepare a 10% bleach solution by mixing 1 part bleach with 9 parts tap water in a plastic or glass container with a lid that will not be used for food storage. Okay, very important. All right, and then you have a video on how to make surface disinfectant. And then you want to soak the bread in the disinfectant bleach solution for 24 hours in a location that's off limits to children and pets. Okay. You want to use enough bleach solution to completely cover and soak the bread. Carefully drain off the excess bleach solution in a sink and dispose of the bread with other household waste. For the brine shrimp should not be released to the environment because they are non-native organisms in many ecosystems. They may be transferred to a glass or plastic container with a lid that will not be used for food storage, along with approximately equal amounts of concentrated bleach. Let the shrimp soak in the bleach for 24 hours before disposing of them, and the bleach down the drain, followed by a generous amount of tap water. A second disposal option is to freeze the shrimp overnight, then pour them down the drain. Okay, so either one of those is the proper disposal for your your activity slides. Okay, very important. And then one thing I do want to mention, if you're unable to take pictures, you can use this sheet to actually draw what you see in the specimen slides. And then on the lines underneath, you're going to label them. Okay, and so there's quite a few of these, so you can use this and add that to your lab report. All right, so if you have any questions, let me know. Enjoy the lab.